welcome everyone for joining us. My name is Harry, and I'm a product specialist in microbial products at Novogene. And the topic of today's webinar is the microbial shotgun metagenomic sequencing, and I will walk you through the entire pipeline of a typical metagenomic approach for your research. That is from how to prepare samples to the popular content of the data analysis nowadays. And I would like to break today's presentation into 4 parts. Firstly, is a brief introduction of what it is of a shotgun metagenomic sequencing. Then is the project workflow from sample preparation to our sequencing. And after that, we will see some popular analysis content in bioinformatics and a case study of an interesting paper. Demonstrating how spaceflight could possibly change microbiota of human being. And last but not least is a comparison between shotgun metagenomics and amplicon-based metagenomics, the two mostly used sequencing techniques in microbial study. And you may find my assistant today, Dr. Novo. So let's first look at it. What is shotgun metagenomic sequencing? These two researchers, Kevin and Liar from University of California, Berkeley, says the shotgun metagenomics is the application of modern genomic techniques, which does not need isolation and lab cultivation of individual microbial species. So we know that our biosphere has 10 to the power of 30 to 10 to the power of 31 microbial genomes, and only less than 1% of them could be cultured. We know that during isolation, there might be some species lost when applying different kinds of species enrichment. And when doing cultivation, it is of high possibility that we would lead in bias due to the culture media preferences. So in this case, since it does not need cultivation and it will not lead in bias by doing shotgun metagenomic sequencing, we could get a more authentic view of the microbial community structure and how those microbes function within the community and the mechanisms of metabolism network, as well as their interactions. And here is the common or standard workflow of a shotgun metagenomic sequencing project. The first step is the sample preparation. And often in metagenomic study, samples are from target environments such as water, feces, gut, or soil. Then the DNA extraction is performed to isolate total DNA from those samples. And at Novogene, we can accept either environmental samples or DNA samples as input. The next step after the total DNA acquisition, we will apply quantification and a DNA quality control to make sure the DNA we have qualifies the standard of a successful library preparation. Then this DNA will be fragmentized to 250 to 300 BP for the construction of a 350 BP insert library by a series of steps like end repair and adapter ligation. And finally, this qualified library will be put on an Illumina NovaSeq 6000 sequencer for data collection and the subsequent bioinformatics analysis. And let's jump into the workflow. And our first step, of course, is sample preparation. So for metagenomic studies, we often take tissues or tissue adjacent samples such as gut, rumen, swabs taken from oral cavity or plant tissues to study endophytes. So in this case, there will be high possibility that the host DNA may be included in the total DNA extracted. the host DNA sequences could take up a large portion of the total DNA obtained, so making the amount of the effective data very low for the subsequent analysis. So we should take measures to avoid the situation from happening. And the first step is that during sampling, we should try not to take the part close to the host tissue. And then when doing extraction, make sure to use the corresponding kit that tailored for different kinds of samples. And also after sequencing, we could try to eliminate host contamination by removing the data aligned to our host reference genome. And for those typical samples which researchers often focus on, we have several suggestions and tips regarding the operation of sampling. So for soil samples, it is recommended to choose the soil which do not directly contact with air. And firstly, we should remove the visible impurities such as roots of plants or tiny pebbles. And then we pass the soil sample through a 2 mm diameter filter to further remove smaller impurities. And for soil samples, it is recommended to collect samples from multi-spots of a location and mix them into one sample. For sludge samples, if we can clearly see water in it, we would recommend to centrifuge the samples at 10,000 rounds per minute for 10 minutes at 4 degrees Celsius first, and then we remove the water. Since in many cases, microbial diversity in water is quite different than that in sludge. And for swabs like the oral cavity swabs, which are often collected now in this period of coronavirus pandemic, we would strongly suggest to swab multiple times to collect sufficient amounts of the microorganism. And commonly, we need 5 to 10 pieces of swabs to meet the requirements of DNA amount in library preparation. And for the water samples, if the waters were turbid with visible impurities, we would recommend to filter them using more than one specific diameters of filters. The ideal condition of the water is transparent with no visible impurities. And last is the gut samples. The gut samples is becoming more and more popular in recent studies. It is highly recommended to avoid touching the mucous membrane and wash the intestine several times with the PBS buffer. Then use a blade to gently scrape off the surface and collect them. This is a very crucial move to avoid host contamination for gut samples. And we know that the replication is a precise process where the exact same findings are re-examined in the same way with identical design, subject selection requirements, and the level of significance as the original research study. And biological replicates are a parallel measurements of biologically distinct samples that capture random biological variation, which may itself be a subject of study or a source of noise. And for shotgun metagenomic sequencing, it is highly recommended to prepare at least 3 biological replicates for both environmental samples and plant or animal samples. And after we completed sample preparation, the next step will be the DNA extraction. And here we have some extraction method recommendations regarding different kinds of sample sources based on our project experiences. And one of the most extensively adopted extraction method is using CTAP solution. Since the polysaccharides and the DNA have different solubilities in CTAP depending on the concentration of sodium chloride. So at higher salt concentrations, the polysaccharides are insoluble, while at lower concentrations, DNA is insoluble. So consequently, by adjusting salt concentration in lysates containing CTAP, the polysaccharide and DNA can be differentially precipitated. And specifically for soil and sludge samples, this power soil DNA isolation kit from Mobile is very effective. And for stool samples, we recommend to use this Kygen KAMP DNA microbiome kit. And after the extraction, the sample amount we need at Novogene is more than 200 nanograms of total DNA, with concentration higher than 10 nanograms per microliter, and the total volume more than 20 microliters. And the next part is when we receive DNA samples, we will perform a sample quality control to make sure they meet our requirements. And basically, we have two criteria. The first one is that when applying the gel electrophoresis to our extracted DNA samples, the main band should be observed above the 500 BP marker. And as we can see from this picture on the right, there is severe degradation and we cannot tell the location of the main band. So it will be labeled as a hold or fail sample, indicating that there may be possibility the following library preparation not successful. And criteria number 2 is about transparency. We should observe no obvious color and the sample should be totally transparent. For example, we could see yellow and red in samples in the pictures number 2 and 3, which are samples of abnormal transparency. This indicates that there might be some problem with the sample that could probably sabotage our subsequent analysis. So in this case, we will continue the process only after we got confirmation from our clients. And generally speaking, however, we do not recommend to continue. And instead, we would suggest clients resending the samples for the better experimental results. And after sample QC, we will move to library preparation. So first, when we got the extracted genomic DNA, we will fragmentize them, perform and repair phosphorylation, A-tailing, and in the next step, we ligate the fragments with the indexed Illumina adapters to go sequencing. And the NovaSeq 6000 is used for shotgun metagenomic sequencing at Novogene, and the strategy we apply is the pair end 150 BP. And after sequencing, we will move to data quality control. And here we label E as the sequencing error rate. Then the Q fret, which is the fret quality, can be written as -10 multiplied by the logarithm of E with base 10. So if the fret quality or fret score is 30, which means the error rate is 1 of 1,000, then the correct rate will be 99.9%, and we call this Q30. And although the sequencing quality will drop during sequencing, or the error rate will increase due to the consumption of reagents, we are still able to guarantee that for a shotgun metagenomic sequencing project at Novogene, more than 80% of the reads could reach Q30, which exceeds the data quality of Illumina's official guarantee of 75%. So for data quality control, one of the important steps is reads filtering. And what we do is that we will filter out 3 kinds of reads. The first one is that the read contains the whole or part of the adapters. And the second one is the read that contains more than 10% of bases that are uncertain after sequencing. And the third one is reads that more than 50% of the bases with fret quality score less than 5. And after these filtration, we will get clean reads for the subsequent analysis. And at Novogene, all read sequences are stored in FASTQ format. And we will include this pie chart in the data QC report, which will show the percentages of the different kind of reads as I have mentioned above. And after data QC, if you chose the bioinformatics package at Novogene, we would continue to the analysis part. So for analysis in metagenome sequencing, we primarily would focus on the answer to these two questions. Who is there in our samples? That is the composition analysis, basically taxonomic diversity and phylogenetic diversity. The other question is, what are they doing? That is how they are functioning in the sample's microbiota. And in our analysis pipeline at Novogene, after the quality control of raw data, we will obtain this clean data. Then the next step is by assembly of reads, the scaftics will be generated to do the following gene prediction. And after we get the non-redundant gene catalog, we will firstly perform taxonomic annotation by using this microNR database. Then we will do the functional annotation by using a CAG database to analyze biological pathways and AgNOC database to do the orthology predictions, and a KZ database to describe the families of the structurally related catalytic and the carbohydrate binding molecules of the enzymes. And after we obtain those annotation results, we can further do pathway analysis as well as some other clustering and the dimension reduced analysis such as PCA, MDS, and PCOA. It's actually the first step in metagenomic bioinformatic analysis is reads assembly. And after pre-processing, the clean data is obtained. Then we use the SOAP de novo assembly software for assembly analysis. For scaftics generated by single sample and mixed assembly, we filter out fragments below 500 BP and perform a statistical analysis and the subsequent gene prediction. So starting from the assembly results, we calculate the distribution of the scaftic length in each sample and plot it into a graph. The results are shown in these two figures. So the first vertical axis here is the frequency, represents the number of the scaftics. And the second vertical axis is the percentage, represents the percentage of the number of scaftics. And the horizontal axis here represents the length of the scaftics. And as we know that biological replication is necessary for any biological experiment, and high throughput sequencing technology is no exception. So the correlation of gene abundance between samples is an important indicator to test the reliability of the experiment and the rationality of sample selection. The closer the correlation coefficient to 1, the higher the similarity of gene abundance patterns between samples. And this is the correlation heat map between samples. So in this figure, different colors represent the level of the Spearman correlation coefficient. And if you see the legend on the right here for the relationship between the correlation coefficient and the color, so the darker the color, the greater the absolute value of the correlation coefficient between samples. And the leftward deviation of this ellipse indicates that the correlation coefficient is negative. And likewise, the right deviated means the coefficient is positive. So, and the flatter the ellipse, the greater the absolute value of the correlation coefficient. And the plot to the right is the box plot of differences in the number of genes between groups. The horizontal axis is the information of each group, and the vertical axis is the number of non-redundant genes. And from this plot, we can see the different level of non-redundant genes in each sample. And in order to investigate the distribution of the number of genes among designated samples or groups, we analyze the common and unique information of genes between different samples or groups. Then we draw a Venn diagram or petal diagram and display the results here. So each circle or petal represents one sample or group, and values in this overlapping parts represents common genes shared by different samples, and the other parts are specific genes in each sample. And for the sake of artistic, it is suggested to use Venn diagram if samples or group number is no more than 5. And if we have more than 5 groups or samples, using petal diagram will be much clearer to see. And for a comprehensive and intuitive display of the relative abundance of species at a different classification level in each sample, we will use this corona display to visualize the results of species annotation. So circle from inside to outside stand for different taxa, and the area of sector means the respective proportion of different taxa. By clicking on different sample names here in this box, we can select the samples which will be displayed on the screen. And if we were interested in a certain keyword, say that we want to know the existence of probacteria in different taxa, we just type in the keywords and click search. And then it will be highlighted on the graph. So here by clicking minus and plus buttons, we can adjust the font size or image size to zoom in or out. And if you click on a species on the graph, we will see on this upper right corner, the percentage of currently the major species that's selected, and the trace species will be displayed here on the lower right corner. And according to the taxonomic annotation results, we select top 10 taxa of each sample or group at each taxonomic rank, that is phylum, class, order, family, or genus, to form the distribution histogram of relative abundance of taxa. So we could visually see the taxa with higher relative abundance and the proportion in different classification levels of each sample. And the relative abundance of taxa in the phylum and genus illustrated in this figure on this page. And y-axis here represents the relative abundance, and x-axis represents the sample name. And others here, this others means a total relative abundance of the rest phyla besides the top 10 phyla we presented above. And according to the abundance information of top 35 genus of our samples, the heat map was drawn so as to check whether the samples with similar processing are clustered or not. And the similarity and difference of samples can also be observed. The result is shown here in this figure. So plotted by sample name on the x-axis, and the y-axis represents the genus, and the absolute value of z here represents the distance between the raw score and the mean of the standard deviation. So the redder the color, the more this phylum clustered in this sample, and vice versa. And at present, the dimensionality reduction analysis suitable for ecological research is mainly this principal component analysis, which is PCA, and this non-metric multidimensional scaling, which is NMDS. And among them, PCA is a dimensionality reduction analysis based on linear model. It uses the methods of variation decomposition to reduce the dimensionality of multi-dimensional data, thereby extracting the most important elements and structures in the data. PCA can extract the two coordinate axes that maximally reflect the difference between samples. So they can tell us the difference of multidimensional data on two-dimensional coordinate graph, and then review the simple laws under the background of complex data. The NMDS is a nonlinear model, and its purpose is to overcome the shortcomings of the linear model and better reflect the nonlinear structure of the ecological data. The NMDS analysis is applied most often to reflect the species information contained in the sample in the form of points in the multi-dimensional spaces. And the degree of difference between different samples is reflected by the distance between the points, so which can reflect the differences between or within group of samples. And based on species abundance table of different classification levels, we conduct PCA and an MDS analysis. If the species composition of the sample is similar, the distance between them in PCA and an MDS map is closer. So for this PCA analysis, each point represents a sample and plotted by a principal component on the x-axis and another principal component on the y-axis. And the points were colored by groups. So the percentage on each axis indicates the contribution value to discrepancy among samples. So the more similar the sample composition, the closer they to each other. And for NMDS analysis, each data point in the graph stands for a sample. The distance between data points reflects the extent of variation, and samples belong to the same group in the same color. When the value of stress factor is less than 0.2, it's considered that an MDS is reliable. So in order to study the similarity of different samples, it is also possible to construct a cluster tree of samples by performing cluster analysis on the samples. And this Bray-Curtis distance is the most commonly used distance indicator in hierarchical clustering. And it is mainly used to describe the similarity between samples, and its size is the main basis for sample classification. So starting from the gene abundance table in each sample, the Bray-Curtis distance matrix is used to perform cluster analysis between samples. So this Bray-Curtis dissimilarity is bounded between 0 and 1, where 0 means the two samples have the same composition, that is, they share all the species. And 1 means the two samples do not share any species. So for example, from the clustering tree here on the left, we can see the Bray-Curtis distance between sample B3 and B1 is about 0.1, while the Bray-Curtis distance between sample A1 and A2 is about 0.4, which is much bigger than 0.1. And we could see that the species composition, the similarity between B3 and B1 is much higher than that between A1 and A2. And the metagenomes provide insight into the physiology of a community by clarifying the collective functions. So those functions are also encoded in the genomes of the organism that make up this community. The functional diversity of a community can be quantified by annotating metagenomic sequences by using several databases. The CAG database is currently developed into a comprehensive database. The core of it is the CAG pathway and the CAG orthology database. And in the CAG orthology database, genes that perform the same function are clustered together, called ortholog groups, also KO entries. So each KO contains multiple gene information and plays a role in one or more pathways. And in the CAG pathway database, biological metabolic pathways are divided into 6 categories. And that is namely the cellular processes, the environmental information processing, the genetic information processing, human disease, metabolism, and the organic system. So each of them is classified into 2, 3, and 4 levels by the system. And the level 2 currently contains 43 seed pathways. The level 3 is its metabolic pathway diagram. And the 4th layer is the specific annotation information for each metabolic pathway diagram. And the eggnog database used alignment algorithm to perform functional annotations on the constructed orthologous groups. And the KZ database is a professional database for studying the carbohydrate enzymes. And for functional annotation graphs on this slide, the number on the bars represents the number of unigenes on the node. And the other axis is the code of each function class of level 1 in each database. So for example, on this CAG pathway annotation graph here to the left, the number on the different colored bars represents the number of unigenes which are annotated to the corresponding functions here to the left. And according to the annotation results of each database, the relative abundance statistics of the functions in each database can be drawn here. And we take the relative abundance statistics of each sample at level 1 in each database as an example. So this vertical axis here represents the relative proportion of annotations to a certain functional category. And the horizontal axis here represents the sample name. And in the metabolic pathway diagram, the nodes here represents various compounds, and lines represents a series of enzyme reactions. The red color here represents enzyme reactions shared by 2 groups or 2 samples, and blue here stand for the unique enzymatic reaction of group A or sample A, and green stand for the unique enzyme reaction in group B or sample B. And whether it is human gut microbes or other environmental microbes, there are many resistance genes. So the abuse of antibiotics leads to irreversible changes in the microbial community, both in the human body and the environment, which poses risk to human health and also the ecological environment. So research on resistance genes has received extensive attention from researchers. And CARD is a newly emerged resistance gene database in recent years. And it has the advantage of the comprehensive information or user friendliness and timely updated and maintenance. And the core component of this database is the antibiotic research ontology, or we call this ARO, which integrates information such as sequence, the antibiotic resistance, and mechanism of action, and associations between AROs, and will provide online interfaces between AROs and PDB and CBI and other databases. So here, this bar plot to the left is the relative abundance of resistance genes, like we have discussed before. And this donut chart here to the right is plotted according to the taxonomic annotation of all the samples in the group, and the proportion of different phyla in all genes and the resistance genes here are shown. Which phylum had more resistance genes can be visually reflected, and the inner circle here is the species distribution of ARO, and the outer circle is the species distribution of genes of all samples in one group. And so far, we have finished introducing the analysis content. And now I would like to share an interesting paper with you. This paper is published on Gut Microbes this year. It is about how spaceflight could influence the function and composition of human gut microbiota. And as we know, the space is a dangerous environment and a risk to human health. And astronaut physiologies change profoundly when exposed to the environment in space. And previous spaceflight studies have shown that the astronauts were easily infected by bacteria or viruses and other pathogens because of the immune dysregulation. So in order to study how will spaceflight influence the composition of human gut microbiota, the author collected fecal samples of astronauts who took spaceflight mission 1 and mission 2. So mission 1 has 3 people and 2 has 2 people. From the fecal samples, they extracted the total DNA and constructed a 350 BP insert DNA library. And then this library was sequenced on a Illumina sequencer by using pair and 150 BP strategy. And by doing also this following analysis, they have some interesting findings that I would like to share with you. Here, the fecal samples were collected from each person at 4 or 5 time points, including 1 week before lunch, that is L-7, and recovered 1 day after spaceflight, which is R+1, recovered 1 week after spaceflight, that is R+7, and 2 weeks after spaceflight, R+14, and 4 weeks after spaceflight, that is R+28. So the author first characterized the gut microbiota composition for mission 1 at the phylum and the genus levels, it is figure A and B. So the gut microbiotas at both the phylum and genus levels fluctuated obviously between R+1 and R+28. And the author then characterized the composition of the gut microbiota for mission 2, and also at phylum and genus levels. That is here from figure C and D. The gut microbiota at both phylum and genus levels also were significantly influenced by the spaceflight. And also the recent studies on microbiology in space have revealed that the spaceflight has an important influence on bacterial resistance genes. So the result of this study showed that the only unique enzyme reactions affected by spaceflight were involved in biosynthesizing antibiotics. So the author analyzed the resistance genes in the gut microbiota from mission 1 and 2. And here, the LEFC analysis was used to obtain the different AROs, and they compared the ARO abundance of R+1 with that of R+28 from mission 1. And the LDA score here of the AROs above 2 and the cluster heat map here are shown. So from the graphs on these 2 slides, the species distribution, species abundance, and resistance genes in the gut microbiotas were both influenced by spaceflight. This is a paper which used a typical shotgun metagenomic sequencing approach that studied and revealed how spaceflight could affect composition of human gut microbiota. And during the communications with our customers, we found that some of them may get confused of whether choosing amplicon sequencing or shotgun metagenomics to study the composition of microbiota. And on this slide, we will show some differences between them in the technical and analysis content aspect. So firstly, in amplicon sequencing, we will amplify a certain target region. For example, 16S V4. This is our region of interest. But in metagenomics, we do not do any amplification to genomic DNA. We just use them directly to fragmentize and to proceed to library preparation. And also we need a relatively smaller data amount in amplicons, commonly 30K to 100K raw reads per sample. But for metagenomics, often we will sequence a large amount of data. We normally choose the 6 gigs package. And for amplicon analysis, we focus on the microbial community structure or composition by doing OTU clustering and diversity analysis. While in metagenomics, although we could also study the composition, and what we care more is genome and its functions by doing gene prediction and annotation based on various databases. And here I made a cartoon that shows the composition, shows the comparison among resequencing. The shotgun metagenomics and amplicon metagenomic sequencing, which are the 3 most common sequencing services you can find on the market. And after we done sampling from water, feces, or soil or gut samples, if we want to study a single bacterium or fungus, we will do culture and separation. And the most common goals are studying the genome composition, a single nucleotide polymorphism, that is the SNP, the insertion or deletion, the indel, and structure variation, the SV. I put parentheses here over copy number variation is because the structural genes of bacteria are always single copy. So for the mutation researchers of the known microbial genomes, we often choose microbial resequencing. And if we want to study the microbial diversity, at the same time, we care more on metabolism pathways of genes, gene functions. We just use total DNA to do the shotgun metagenomic sequencing. And if all we care is species identification and diversity, commonly we will choose an amplicon sequencing, and it's the most cost-effective approach. So with all that being said, what makes us different from other companies in the market? Well, the most important one is that we have a vast majority of experiences in metagenomics and microbial sequencing. Our clients had published various papers on related journals, and up to the end of 2019, the total count of published papers by our customers have reached 100, with a total IF of 620. And I'm very proud to say that Novogene has announced Falcon, this fully automatic intelligent platform that could integrate all pre-sequencing processes from extraction to library quality control. And in this way, Falcon greatly shortened the project turnaround time by processing 2,000 to 3,000 samples in just 24 hours, which could lead to a 60% decrease of production time. And thanks to our world-leading computing and server capabilities, we could carry a vast number of projects in parallel, and at the same time, still achieving amazing data stability and turnaround time. And also, we are very proud that we have more than 2,600 of collaborative research organizations and universities all around the world, and more than 1,200 collaborative companies, and more than 590 hospitals. We are proud of so many partners working and doing research with us, and we hope you could be one too. So this brings us to the end of the presentation, and thank you for joining us. And also, I would like to thank our fantastic panelist group, Manoj, CB, Mary, and Chloe, for supporting today's webinar. And if you would like more information, please do not hesitate to see our website at en.novogene.com or send us email at inquiry_us@novogene.com. For customers who speak Spanish or Portuguese, we also have a dedicated sales manager, Dr. Diego Levi, who is now based in Argentina. And please contact us for any information, and we are responsive and glad to help at any times. So now I'm more than happy to take questions. You may let us know your questions by the Q&A function in Zoom.
Thank you, Harry, for that webinar and for helping us with that. As Harry mentioned, we are entering the live Q&A part of our webinar. Please use the Q&A module to post your questions. We have a few questions coming in. Give us a second to process that, and we will be answering a few of those. Just a quick reminder, we will be sending out the slides and a recording of this webinar to you using the email that you registered with. So any questions that we're not able to answer, since we only have a few minutes left, we will answer this via email directly to you, and we will make this available on our website as well. So we'll include all those details in the follow-up email. So I do see a few questions coming in. Let me take one of these. So there's a question about from an anonymous attendee. Is Novogene bioinformatics software available to customers? Harry, could you speak to that a little bit more, just to give clients a perspective on whether or not they have access to our bioinformatics pipeline?
Okay, thank you for your question. And currently, Novogene bioinformatics software is not available to our customers, but we do have some cloud platforms that our customers who chose to do analysis at Novogene can log onto and do some custom analysis using our cloud platform, and we have our analysis softwares plugged in. So maybe this is one of the way that you could use the analysis software we are now using.
Awesome. And we had another one regarding recommendations for samples that we suspect is degraded. I think that our panelists had answered that, but Harry, would you like to elaborate on that a little bit more?
Sure. This is also a very good question. And honestly speaking, that this really depends on how degraded the sample is. And after our sample QC, if we can see the main band clearly, there are some short fragments, and I recommend we filter out the short fragments and increase the sequencing data amount to get enough data. But if we cannot find the main band, based on the project experiences we have, I would suggest try risky library preparation for 1 or 2 samples and see how the result goes.
Awesome. So we have time for one more question. Again, the questions that were asked that are coming in, we will reply via email after this webinar. So I'm going to take one more question. There's a question on mapping versus assembly in metagenomics. Harry, would you mind speaking to that a little bit more?
Sure. Yes, this is a very good question. And now we have basically 2 methods for the metagenomics analysis. First one is the mapping. The second one is doing it by assembly. And mapping is also called the read-based metagenomics analysis. And it basically used unassembled reads. And it was actually one of the first method to be used, but it is still valuable for the quantitative analysis, especially if we want to do the research of relevant references and the references available. But for the assembly-based metagenomics, this workflow is attempt to assemble the reads from one or more samples, or to bin context from the samples into genomes, then analyze the genes and context. So the assembly-based metagenomics is a more precise one, and the mapping-based is a more cost-effective one, and is tailored for the quantitative analysis.
Awesome. Well, that kind of wraps up our time together. Again, any question that we were not able to answer, we will answer in our follow-up email. Thank you again, everyone that attended. We appreciate you joining us. Stay tuned for more webinars. Again, please follow us on social media to stay tuned with our next webinar series, promotions, and any sort of other content that we're putting out there to help inform and educate you. Thank you again. Have a great day.